Hello friends, from today we are start online teaching of BSc Part 2, semester 4th, subject Botany, paper number 8. So in that the unit enzyme is allotted to me, and therefore we have to discuss about the introduction of enzyme, classification of the enzyme, nomenclature, then action of the enzyme, properties, structure, as well as the effect of temperature and pH on enzyme activity. So all these things we have studied in this unit. So first we go with the introduction of the enzyme. So the enzyme is the term coming from the Greek word. There is a two terms. The first part there is en, that is means in. Enzyme is nothing but yeast. So the enzyme is the protein which is present in the yeast, and the yeast have the ability to ferment the sugar into alcohol and carbon dioxide. Therefore, the name enzyme is given to this protein. This enzyme or enzymes are the catalyst formed in the cell for specific chemical reaction. In general, in chemistry, we call it as a catalyst, but here this chemical protein or this enzyme is synthesized in the living cell for specific chemical reaction. Therefore, here we have call it as a biocatalyst or we call it as an enzyme. In biological system, the reactions are catalyzed by special and remarkable protein. So in botany as well as in zoology, you have no different processes are there. For example, the photosynthesis, glycolysis. So in that there are series of the reactions, and each and every reaction is complete or it is catalyzed by a special and remarkable protein which are present in the cell. This is called as a enzyme, and therefore this enzyme are called as a biocatalyst. That means the biocatalyst or this enzyme, they accelerate the rate of biochemical reaction in a proper condition of temperature and pH. The enzyme can be defined as, another definition of the enzyme is a complex chemical protein produced in living cell. So enzyme is the complex chemical protein which are produced in the living cell which catalyze a biochemical reaction at very low concentration of temperature and pH are called as an enzyme. In short, the enzyme is a biocatalyst which accelerates the rate of reaction or rate of biochemical reaction. Therefore, we say that all enzymes are protein. That means the enzyme is always protein, but all proteins are not enzyme. You have to remember that each and every enzyme is a protein, but all protein are not an enzyme. Enzymes are important practical tool in medicine, chemical industry, food industry and agriculture. So regarding the discovery of the enzyme, in 1860 the Louis Pasteur is the well-known scientist, microbiologist, recognized the potentiality of yeast cell to ferment the sugar. But he cannot discover the enzyme. He only stated that there is something in the yeast which have the ability to ferment the sugar into the alcohol and carbon dioxide. The term enzyme was first coined by Frederick Cuny, so in 1878. Then scientist Booker in 1897 first made the extract of the enzyme. So he first made the enzyme, and the scientist Davis Summer in 1926 isolated and purified the enzyme urease and conclude that enzymes are the protein. So here this is in that the scientist Frederick Cuny which gives the term enzyme first time, and the Davis Summer is the first time isolate the urease enzyme, and he conclude that the enzyme are the protein. Then there are more than 200 different enzymes are there, and therefore there is need to be classified the enzyme. for study purpose, and therefore the next point in your unit, that is the classification and nomenclature of enzyme. For systemic arrangement, classification and nomenclature of different enzymes, in 1962 established a commission called as a International Union of Biochemistry. In short, it is called as IUB, International Union of Biochemistry. The commission was revised in 1964, and the commission formulated certain rules and regulation for nomenclature and classification of the enzyme, because the number is enormous and this number we cannot remember at a time. Therefore, there is a need of the classification nomenclature of the enzyme. So first we studied about the rules of the nomenclature, how we can give the name to the enzyme, because the number is large number, and therefore there is the essentiality to give the scientific name to the enzyme. So this is some regulation or rules of the nomenclature of the enzyme. So most of the enzymes are named by adding suffix ase to name to the substrate. So here first the rule states that you have to give the name of the substrate on which enzyme acts, and you have to add the ase as a suffix. For example, urea is the substrate. You have to add ase as the suffix. The name of the enzyme becomes urease. Similarly, there is also lipase. That means ase is the suffix of the to the substrate. Then second, sometimes the enzymes are named on the basis of reaction that they are catalyzed. Which type of reaction the enzyme catalyzes? So such type of reaction you have to give the name. For example, dehydrogenation, that means the removal of hydrogen atom, is the function of the enzyme, and therefore the scientific we can give the name as a dehydrogenase, as well as for example decarboxylase. So this is another example of the this second rule. The third rule, the name of enzyme consists of two parts. The first part denotes the name of substrate on which the enzyme acts. You have no, enzyme is always specific for the substrate. It always acts on the specific substrate. Therefore, for giving at the time of giving the name of the enzyme should have the two parts. The first part is a name of substrate, while the second part indicate the type of action of enzyme. For example, here the isocytric dehydrogenase. So the isocytric is the first part, dehydrogenase is the second part. The isocytric is a name of substrate, and dehydrogenase is a type of reaction or type of reaction. The enzyme acts upon isocytric and remove hydrogen from it. Therefore, we can give the name as a isocytric dehydrogenase. Number next, some enzymes have been named based on source from which they were first identified. For example, papain. So the papain enzyme is isolated from the papaya. Therefore, the scientists give the name as a papain. The next one, the name of the enzyme ends with in, indicating that they are basically protein. There are some enzyme, they are ends with the in. So this is also one more rule. For example, the pepsin. The pepsin is at the end there is in, that means it is a protein. Trypsin. So here this is also the one more regulation of the nomenclature. So here in this way we can give the name to the enzyme in the biochemistry. Then classification of the enzyme. As we have known, there is thousands of the enzymes are discovered by different scientists. Therefore, they are classified on the specific manner. So the IUB classify all the enzymes into six major class. Each major class is again divided into subclass. Then subclass is again divided into subclass, and the subclass is again divided into subclass. So here this is the way of the classification of enzyme. As well as there is another one rule. Each and every enzyme, they have a 4-digit code symbol, and this digit code is called as a EC, that means enzyme code. So that means each and every enzyme, they have the 4-digit code. If you take the example of this one here, that is example oxidase. So this oxidase enzyme, they have the 4-digit code 1, 2, 3 and 4. So these 4 numbers are there. The first number indicates the major class, second number indicates the subclass, third number indicates the subclass, and the fourth number indicates the serial number of identity particular enzyme within the subclass. So here this is the enzyme code. That means the first indicate the major class, this is indicate the subclass, the third indicate the subclass, and fourth fourth digit indicates the series of the serial number of that enzyme in particular subclass. So here we can elaborate by taking one more example, that is oxidoreductase. So here the enzyme oxidoreductase. So here the name is oxidoreductase, the enzyme is oxidase. So here EC 1, 2, 3, 4. Again we have to explain here. EC means enzyme code. So first indicate the name of class, that is the major class, class number 1, which is oxidoreductase. Second name, second digit, subclass 2 acts on the aldehyde or oxo group of donor. Third number indicate the subclass number 3, oxygen is an acceptor, and fourth digit indicate the serial number of the subclass, that is the fourth enzyme classified in this subclass. That means here this is the enzyme code, and on the basis of this enzyme code we can identify the type of the enzyme, name of enzyme, class and subclass of the enzyme. Then the next point, that is we can detail study the major class of the enzyme. Already we all know there is 6 major classes are there. That means all enzymes are classified into these 6 major classes. So first is oxidoreductase, second transferase, third hydrolase, fourth lyase, fifth isomerase, and sixth ligase or synthetase. So here this is the 6 major classes of the enzyme. So one by one we have to discuss what is meant by oxidoreductase, what is meant by all these classes with the example. So here the first class, class number 1, oxidoreductase. So the name indicates that there is a oxidation and reduction reaction are catalyzed by the enzyme. All such enzymes are included under class first oxidoreductase. So this enzyme catalyzes oxidation and reduction reaction. In this group, one compound is oxidized and another compound is reduced. They act by transferring electron and hydrogen ions. For example, oxidase, dehydrogenase, reductase, hydroxylase, peroxidase, catalysis, oxygenase. So all these enzymes are grouped into oxidoreductase. And if you take the example of one reaction, that is NAD, nicotine amide dinucleotide oxidoreductase. So in that the substrate is alcohol. This NAD, which is acts as a coenzyme, and this is reversible reaction which form the aldehyde and NADH2. So this is the enzyme lactase, is again NAD converted into pyruvate and NAD. So here this is the oxidoreductase. In that there is one compound is oxidized, another compound is reduced. So this class is called as oxidoreductase. Second class is a transferase. So in this transferase class, the enzymes are included in this class which are involved in transferring the functional group between a donor and acceptor molecule. For example, the group formyl, glycol, methyl, carboxyl, acetyl, phosphate. So from donor to acceptor. So the example of this transferase is a glucokinase. So in glucokinase, adenosine triphosphate plus D-glucose, they are converted into ADP, and D-glucose 6-phosphate. So here that means here there is a transfer of the group, and the important group in this class are aminotransferase, then it is called as transfer amino group. When it transfer the phosphoryl group from ATP, then it is a kinase. When it is transfer aldol group, then it is a trans aldolase, and when it is a transfer ketone group, then it is called as a trans ketolase. So here this is the second class of the enzyme where the groups are transfer between donor and acceptor molecule. So here this is the role of the enzyme. Third class is hydrolase. So this hydrolase class is a special type of transferase where donor group transfer to water molecule. So the name indicates hydrolase, the transfer to the water molecule. The enzymatic reaction is hydrolytic cleavage, that is splitting of the hydrogen. This enzyme split CO group bond C single bond N, C single bond C, O single bond P, and C single bond S. For example, here ATPase. So the ATPase plus H2O converted into ADP, adenosine diphosphate, and inorganic phosphate here. Then here the lipase, that is the fat plus water, is converted into fatty acid and glycerol. The important type of hydrolysis are lipase, peptidase, phosphatases, amidase, glycosidase, ribonuclease, and deaminase. So this is the hydrolysis group. So in that there is a transfer of the group to the water molecule. Fourth class, the enzyme of this group catalyze the addition of group to double bond or catalyzed by electronic rearrangement of the molecule. So the enzymes acts on this bond as well as halide bond. The example is a decarboxylase, in which it removes CO2 from keto acid. Second one is fructose diphosphate aldolase enzyme. So this fructose 1,6-diphosphate is catalyzed into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So here this is the number 4, the lyase. Number 5, the isomerases. That is isomerization reaction is catalyzed by this enzyme. And due to isomerization reaction, there is intramolecular rearrangement of atom in a substrate. It includes cis-trans ketoenol and aldose ketose interconversion. That means cis into trans, trans into cis, keto into enol, enol into keto. So here there is an interconversion and includes the epimerase and some metase enzyme. So the epimerase enzyme is acts on D-xylulose 5-phosphate and it converts into D-ribulose 5-phosphate. So this is the number 5th isomerase class. Last 6th major class is a ligase, or it is called as a synthetase. So this enzyme catalyze bond formation and are associated with the hydrolysis of ATP or other nucleotide phosphate. So here the well-known example is a glutamine synthetase enzyme. It acts on glutamate and NH3 plus ATP converted into glutamine plus ADP plus inorganic phosphate and the water molecule. So here this is the ligase or the synthetase enzyme. This is also called as joining enzyme in DNA replication. Then the structure of the enzyme. So the enzyme actually it is the protein. All enzymes are protein. Therefore it is made up of one or more polypeptide chain. The proteins are nothing but the building blocks of the amino acid. That means the monomer of the amino acids are joined together, form a polypeptide chain. These chains are made up of number of amino acids and linked by polypeptide bond. The sequence of amino acid in polypeptide chain is very specific for a particular enzyme and it is differ in different enzyme. Each molecule, like any other protein molecule, shows the structure, different structure, the primary structure, secondary structure, tertiary structure and quaternary structure of the enzyme or the protein. So here the primary structure means simple protein. There is a simple sequence of amino acid connected to the peptide bond in polypeptide. Suppose this is the polypeptide chain and here this granular structure are called as amino acid. They are joined together by peptide bond, and this simple protein is nothing but the primary structure of the protein or primary structure of the enzyme. Second structure is nothing but it involves the specific folding in long polypeptide chain which is stabilized by synthesis of hydrogen bond between amide and carboxyl group in the amino acid. The common folding are alpha and beta folding, and here there is a development of hydrogen bond and stabilizing the folding. So here this structure is called as secondary structure, which is slightly complex than the primary structure of the protein. This is a tertiary structure, and in this structure is the way in which the helical and random region folds with respect with each other. The tertiary structure gives the enzyme molecule three-dimensional structure. So this tertiary structure is complex than the secondary structure, which is a three-dimensional structure, which is maintained by formation of disulfide and hydrogen bond. In tertiary structure, there is a disulfide and hydrogen bonds are there. There is also presence of ionic interaction, hydrophobic interaction, when their walls forces already we have studied in first year. In the development of these interactions are useful in development of the tertiary structure. The tertiary structure refers to the three-dimensional relationship of the amino acid, that means alpha sheets and beta sheets. So collectively they form tertiary structure of the protein. And then this is the quaternary structure, which is spatial structure, because there is a spatial arrangement of various tertiary structures. Various tertiary structures are comes together and it gives rise to the quaternary structure. Some of the proteins are composed of two or more polypeptide chain referred to as a subunit. The spatial arrangement of these subunits with respect of each other is known as a quaternary structure. This quaternary structure is very much complex structure. So here in this lecture we have to discuss about what is meant by enzyme, then different nomenclature of the enzyme, the rules of nomenclature, classification of the nomenclature, major classes, enzyme code, that is a 4-digit code, 6 major class of the enzyme, and properties of the structure of the enzyme, that is a primary structure, secondary structure, tertiary structure, and quaternary structure.